So this is part 7 of This is a Record of our lecture for Chapter 5. And hopefully this is the last one. But we left off talking about fatty acids and glycerol combining to make a lipid. And so if we have 3 fatty acids attached to the glycerol molecule, then we have a triglyceride. If you only have 2, you have a diglyceride. And if you only have 1, you have a monoglyceride. So those are terms that you'll see on food packaging. So now you'll know what they mean if you didn't know before. And a fatty acid, again, consists of a carboxyl group that allows it to attach to the glycerol. And next to that carboxyl group is a long carbon skeleton that varies from lipid to lipid. But it's often 16 to 18 carbons long. And these nonpolar carbon-hydrogen bonds in this long hydrocarbon skeleton are what make fats hydrophobic. So, for example, why if you put a fat in water, it'll bead up and not dissolve in the water. It is hydrophobic because of these fatty acid chains. Okay, the other term that you will see on food packaging is saturated or unsaturated fats. And so the difference you need to be aware of in review, a saturated fat is when the fatty acid is a long straight chain, and it's because all the carbons, all the valences are filled with hydrogens, and there are no double bonds between carbons. So that allows the formation and structure of the fatty acid to be long, straight chains. So most animal fats are saturated, and that's why they're solid at room temperature, because they are able to pack closely together because of the long, straight length of these chains. You can just think of if you're stacking sheets of paper that are flat, you can get a lot more in a single space than if they're crumpled up. You're not going to get as many. Okay, I'm just making sure we covered everything. So unsaturated fats, in comparison, are like the crumpled paper. And that's because in the fatty acid chain, you end up with some carbon-carbon double bonds. And so what that means in contrast to the saturated fat is instead of having every carbon's valence filled with a hydrogen, and we have one less hydrogen that is then allowing there to be a double bond between the carbons in the skeleton. So these are the things that are the liquids at room temperature, plant and fish fats, vegetable oils. The kinks that are made by the double bonds prevent these molecules from packing close together. So when it says monounsaturated or polyunsaturated, what does that mean? So the difference between a mono and a polyunsaturated fat has to do with how many double bonds are actually present. So in poly, there are many unsaturated chemical bonds, and in mono, there's only one unsaturated chemical bond. So what that causes to occur is that polyunsaturated oils are going to remain liquid at room temperature and in the refrigerator. So as you cool them down, they'll still stay in that liquid form. And so things like safflower, sesame, sunflower oils, those are polyunsaturated. But monounsaturated oils will be liquid at room temperature, but if you put them in the refrigerator, they'll start to solidify. So examples of monounsaturated oils are canola oil, olive oil, and peanut oil. Avocados are also monounsaturated. So these oils, again, if you put them in the refrigerator, they get much more hardened. So you can, if you, I'm sure, have eaten peanut butter or are familiar with peanut butter, if you put it in the refrigerator, it can be really hard to spread when you put it out. So, but if it's at room temperature, it just easily smooths onto a piece of bread. So that's a good example of this. So mostly these are plant lipids, the unsaturated fats, but they actually cause a lot of problems for companies, food manufacturers, because they want to make their product easier to use. So they want to keep the oil solid at room temperature. And in order to do that, they hydrogenate it, which means they chemically alter it so that it goes from unsaturated to saturated. So a hydrogenated fat or hydrogenated vegetable oil means that the unsaturated fats have been synthetically converted to saturated fat by simply adding hydrogens. So it breaks those double bonds and it replaces the double bond between carbon atoms to one that is filled with hydrogens. So this is an example here. And it's done by infusing the substance, the oil, with a hydrogen gas and heat. And they also use a nickel as a catalyst. So in this, they have a mineral catalyst instead of an enzyme that is able to facilitate this reaction. Let's see. Oh, okay. The other thing I wanted to bring up, so this is actually showing you partially hydrogenated vegetable oil simply means that only part of the fatty acid chain has been hydrogenated. It's not completely. But one of the things that is significant about hydrogenated fats is their health significance. So what does it do nutritionally to us as we consume them? And you can see there's so many advertisements for things that are non-hydrogenated, and it really is considered a good practice not to eat hydrogenated fats. And the reason you can see here trans fat free is because when fats are hydrogenated, it changes the configuration of the fatty acid from a cis configuration to a trans configuration. So as you recall with isomers, cis means the attachments are on the same side, and trans means they're across from each other. And so when it says trans fat free, what it's saying is through this process, the isomers are created of this naturally occurring unsaturated liquid oil. And so here's the cis form, and then this is the trans form. And as we learned with isomers in the previous chapter, changing the configuration of a molecule changes its function and how it reacts in an organism. And so just even the simple change from cis to trans can have a significant change in how the fat is processed in the body. Okay, so another important characteristic or example of fats and lipids are the phospholipids. And as you remember from regular bio, those are what make up our cell membranes. And the important characteristic of a phospholipid is that it actually is both hydrophobic and hydrophilic. So a phospholipid takes a lipid portion, its fatty acid tail, which has the hydrophobic and lipid character of the molecule, and combines it with a phosphate group. So the head of a phospholipid is filled with oxygens, which are electronegative and very hydrophilic. So it makes a phospholipid lipid have a split personality, or it's amphipathic, meaning it has both characteristics of hydrophobic and hydrophilic. So here again, the structure is glycerol with two fatty acids and a phosphate. And that phosphate group gives a negative charge to the molecule. So we end up with this hydrophilic head, which is attracted to water, and the hydrophobic tail, which hides from water. So if you put a phospholipid into water, what it will do is self-assemble into what is called a micelle. This is a micelle where the outside is the hydrophilic portion and the inside is the hydrophobic tails, which all are going to try and get away from water. And the same thing with our double layered membrane, bilayer membrane in cells. We have the hydrophilic portion facing the water outside the cell, and the hydrophilic portion, the phosphate head, facing the inside of the cell, which is also aqueous or water. And then the inside of the cell membrane is the hydrophobic portion, which is again trying to get away from the water. And as we'll study this more carefully in our next unit with cells, this has tremendous functional qualities simply because of this hydrophilic hydrophobic configuration. One of those is the fact that it allows the phospholipids to create a barrier for the cell. Okay, another type of lipid or fat are steroids. So steroids are hormones, and they are very different looking fats. And they're another type of steroid is cholesterol. So again, these are considered part of the lipid family, but their configuration includes 4 fused carbon rings and then something attached to those. So the different steroids are created by attaching different functional groups to these rings. And again, the different structure is going to create different functions. So this is a picture or a diagram of cholesterol. So you can see its configuration has the 4 fused carbon rings with just 4 attachments here. And we talked previously about the difference between estradiol and testosterone. Again, here's our 4 fused rings of the steroid, and the only difference between estrogen and testosterone is just these 3 functional groups. So in this case, this carbon has a hydroxyl, this one has a carbonyl, and then there's an additional CH2 or CH3 actually that is attached at this carbon right here. Cholesterol, which is part of this steroid group, is an important cell component. It's part of the cell membrane and helps actually provide structure and strength to the cell membrane. It's also a precursor to all other steroids, including the sex hormones. But it also has negative effects. If you have too much of it in your blood, it can contribute to cardiovascular disease. So you can see here the different examples of steroid hormones that are going to derive from cholesterol. Okay, so the last thing I want to talk about in this lecture are isomers. So I want to just go over this one more time. So there are just a few different isomer formations that you should be familiar with. One is the structural isomer, which just means you have a different molecular structure. The next, oops, these are questions. I don't really want to ask those questions. Oh goodness, it just has the one there. Okay, well, I think I'll go to the questions then. Which term correctly describes the relationship between these two sugar molecules? And you should see that this is a structural isomer because we have different bonds occurring. Okay, and these are questions that are not about isomers. So I'm going to end the lecture right here because it looks like my